In this video, you will learn how to pipette a plate for the PCR reaction. To begin, take out a plastic tray from the drawer and tape it to the lab bench. Now, take a blue support base and snap it securely onto the tray. Now, take a 96 well reaction plate out of the box and place it into the blue support base. Using a Sharpie, you can make small marks on the plate to help you remember where certain genes and samples are supposed to go. Now we will take a look at how to pipette the master mixes. Always pipette from left to right, first going across the rows and then down the columns for each given gene. This will help you to prevent contamination. In this case, the leftmost master mix is 18S RNA. So, we will first pipette 15 microliters of this into the top leftmost well, A1. Always pipette onto the side of the well. For master mixes, pipette to the bottom left. Later, when you pipette the samples, pipette to the top right. This will allow you to reuse the same pipette tip, because the pipette will never have touched the other liquid in the well. It is of utmost importance to make sure that you get all of the sample out of the pipette tip, so make sure to always check the pipette tip for any excess liquid. Continue this procedure for all additional master mixes. When you are not using a given master mix, let it rest on ice. When you are done, you should be able to see all of the master mix droplets inside of the wells. You are now done pipetting the master mixes. We will now take a look at pipetting the cDNA samples. When pipetting the cDNA samples, always move from left to right and from top to bottom on the plate. It is important to note that there are different dilutions used for 18S and all of the other genes. Because 18S uses the 1:160 dilution, you should pipette this first. Then, using the 1:5 dilution, you can pipette the rest of the samples for the remaining genes. Always pipette the PCR blank last. To begin, select sample A in the 1:160 dilution. You will use this for the 18S wells. Pipette 5 microliters of this dilution into the upper right-hand corner of well A1. Then, as long as you have not touched the master mix with the pipette tip, use the same tip to pipette into A2. Now, change pipette tips and move on to sample B. When you have finished the 1:160 dilutions for 18S, you can begin to pipette using the 1:5 dilutions for the remaining genes. When pipetting the 1:5 dilutions, pipette across the entire row, regardless of master mix. However, as before, if you touch the master mix with the pipette tip, make sure to get a new tip. Complete this for all samples. Now, pipette 5 microliters of molecular biograde water into the PCR blank wells. When doing this, be extra careful not to touch the master mix in the wells. If you do, change the pipette tip. You have now finished pipetting the plate. Take an optical adhesive cover out of the box and get the gray beveled tool from the drawer. Gently tap the plate on the bench top to move all droplets to the bottom of the wells. Attach the cover to the top of the plate. Use the gray beveled tool to firmly secure the cover above all of the wells. Tear off the edges of the cover along the perforations. Take the plate over to the centrifuge and place it in the specially designed cradle. Then, close the lid and set the speed to 3,500 RPM, the time to 5 minutes, and the temperature to 4 degrees C. Press the play button to start the centrifuge. When the cycle is over, open the centrifuge. Check the plate for bubbles. Bubbles can cause the machine to incorrectly read the amount of fluorescence in the sample. If there are bubbles present, you will need to repeat the centrifugation. If not, you are ready to put the sample into the machine. To learn how to set up the computer, please watch the next video.